Bismillahirrahmanirrahim. 9700 paper 13 November 23. O Lord, give us in this world that which is good, and in the hereafter that which is good, and save us from the torment of the fire. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. November 23, paper 13, and this is 1 hour and 15 minutes, which is going to be the new timing since June 22 that we have the syllabus changed. Now, coming to question number 1, which statement about light microscopy is correct? As the resolution increases, the magnification increases, not necessarily. As the magnification increases, the image always becomes clearer. No, of sudden it becomes blurred. And we talked about light microscope. The most useful magnification is x1500. After that, if you can magnify it, you can just change the lenses and magnify it, but then it becomes blurred and you cannot see the details. You cannot see the details. Don't say clear. Yes, C is correct that the resolution will decrease as colored light of increasing wavelength is used. You see, wavelength, it has to be, if anything is lesser than half the wavelength, then you cannot see it. It has to more than half the wavelength. So say half the wavelength is 200 nanometers. So you can see objects bigger than 200 nanometers. Now, if the wavelength increases to say 400, 600, then half the wavelength will be 300. So then you'll have to see objects bigger than 300. Now going back to from the book, I'll just explain this once again. You see here in this diagram, you can see wavelength of light is 400 nanometers. So the ribosomes which are 25 nanometers, you cannot see them. It says very clearly, if an object is any smaller than half the wavelength of the radiation used to view it, it cannot be seen separately from nearby objects. So if the wavelength was 400, so you would see things bigger than half the wavelength would be 200. But if the wavelength was 600, then you would see things which are bigger than 300. So we're talking of nanometers. So this is the reason why the part C of the question is right. The diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer is 0.1 millimeter. So this is 0.1 millimeter. 0.1 millimeter. The stage micrometer is replaced by a slide of a plant cell. What is the maximum actual length of the nucleus? Now this is the nucleus. So now you need to see this. And then you have to tell me the actual length of the nucleus according to that. Question number 2. The diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer is 0.1 millimeter. So 0.1, so 40, you can see 50 to 90 is 40. 40 is equal to 0.1, so 1 is equal to 0.1. Now we know this, from here to here you're going to see the nucleus. This is the nucleus. Then you're going to see this, and this is equal to 10. So 0.1 over 40 into 1000. Why have I done this to 1000? Because this is millimeter, and 1 millimeter is equal to 1000 micrometer. So multiply and you get it. 1 is equal to 2.5 micrometer, but these are 10 divisions. You can say these are 10 divisions. So 10 into 2.5 is 25 micrometer. So the answer is B. Question number 3. What is the correct order for the involvement of the cell structures in the synthesis and secretion of an enzyme? Cell surface membrane, Golgi, nucleus, RER. Now we all know that the nucleus is the first thing. So 3 would be the first one. Nucleus is the first thing, or involvement of the cell structures. So in a secretion of an enzyme, in a protein first there has to be transcription. Now that can only take place in the nucleus. There was only one possibility starting from 3. The rest are all wrong, because you see in the nucleus transcription has to take place, and then the mRNA is formed, and then the mRNA moves and then attaches to the ribosome on the rough endoplasmic reticulum. So nucleus first, then rough endoplasmic reticulum, then goes to the Golgi and is modified, and then goes to the cell surface membrane for exocytosis, secretion of an enzyme. So that means it's made in the cell, but then it is thrown out, secreted out, like amylase made in the cells and thrown out into the saliva. The saliva takes it away to the mouth. Salivary glands make saliva and contains an enzyme called amylase. Next, question number 4. Centrioles, cilia, microtubules, and microvilli are structures found in eukaryotic cells. Which statement about these cell structures is correct? Centrioles, cilia, and microvilli are composed of, no, I'm sorry, cilia are not composed, and microvilli are not composed of microtubules. Centrioles and cilia are. Microvilli are what? They're just surface foldings. The cell membrane is folded, microvilli. Centrioles play a role in mitosis and semi-conservative DNA replication. No. DNA replication, no centrioles take part in S phase of interphase. Then cilia and microvilli both increase the surface area. No. The cilia, which are found in the ciliated epithelium cells in the trachea, well, they're just going to sweep. They don't increase the surface area. They just sweep the mucus upwards. So the answer is D. Others are all wrong, because cilia do not increase the surface area for absorption. Cilia just sweep the mucus upwards towards the throat, where it is then swallowed. Then question number 5. Which description of plasmodesmata is correct? Now let's read all of them and see which one is right. They are channels through plant cell walls lined by the cell membrane. Yes, that is right. So the answer is A. Let's look at the let's do a little bit of clarity of thought. This is right, 2 cells. So this is the cell wall of this one, this is the cell wall of this one, and then this is the cell wall of this one. And then we have, like you see, this has 3 lines, and then we have a plasmodesmata. Okay, so let me now show you how this plasmodesmata. So the plasmodesmata is, this is the cell membrane which I'm drawing in yellow. And then this is the plasma. This is the cell membrane. It goes all around it, and then it goes here. So when water passes from this cell to this cell, actually does not cross any partially permeable membrane. This is not osmosis. It says they are channels through plant cell walls lined by cell surface membrane. Well, if you didn't know this, that's sad, very sad. Then it says they are channels through plant cell walls that are formed from proteins. This is wrong. It's not made of proteins. They are channels required for the movement of water through the apoplast. Apoplast is along the cell wall. It's not from cytoplasm to cytoplasm. That's a symplast pathway. They can become lignified and form pits in the wall of the xylem vessel element. No, that's wrong as well. So plasmodesmata is not that. The plasmodesmata, when the lignin is deposited, then the areas where there was plasmodesmata, they form the pits. So they don't become lignified. There is no lignin there, in fact, in the pits. Then question number 6. Which organelle does not have nucleic acids? Very easy, Golgi body. Chloroplast has circular DNA, mitochondria has circular DNA, ribosomes has ribosomal RNA. And that RNA is a nucleic acid. Nucleic acids are DNA and RNA. Now, which processes occur in eukaryotes and prokaryotes? Now, you know the first thing, mitosis does not take place in prokaryotes. What takes place in prokaryotes is binary fission. And binary fission is nothing like, it's not mitosis, it's just a bacteria has a circular DNA, then another DNA forms, and this bacteria divides into 2. That's called binary fission. It's not mitosis. So anything without 2 was correct. Now you know this is 2, this is 2, this is 2. So only this is correct. Anything with 2 was incorrect. But if you knew that, I mean, if you are really going to not know that, then I'm sad. Question number 8. Which bonds are present in all viruses? Which bonds are present in all viruses? 1 and 3. Ester bonds are not formed because every virus does not have a fat. So that is why question number 8 The answer is C, 1 and 3 only. Covalent bond and phosphodiester bond. Phosphodiester bond would be between the nucleotides, and covalent bond would be the peptide bond on protein would be a covalent bond. Question number 9. The diagram shows the structure of a monomer. Which molecules contain the monomer? Amylopectin, glycogen, sucrose. So this is either alpha glucose or beta glucose. Now how are you going to figure this out? Let me just make you remember it. Now looking at this diagram, now look at the mnemonic that I've made for this. Alpha below, beta above. So alpha below, beta above. Where is it above? On carbon 1, the OH is below, and in beta the OH is above. So this is just a way of you to remember it. And now let's go back to that. So you can say here it is alpha is below. So this is alpha glucose. Now alpha glucose is present in amylopectin, sucrose and glycogen. Beta is only present in cellulose, ABC. B is only present cellulose only. So the answer is B because it's present in all the three of them. A triglyceride consists of glycerol and three different fatty acids L, O and P. The diagram shows one possible arrangement LOP. I say what is the total number of different arrangements of the fatty acids? So it can be OLP, POL, OPL. So the answer is A is 3. Then coming to question number 11. Question number 11, which statement about phospholipid molecules is correct? They contain one saturated and two unsaturated. A phosphor molecule only contains two fatty acids. Phosphodiester bonds are in DNA or RNA. Hydrophobic and hydrophilic regions. They contain hydrophobic, and yes, that is correct. C is correct. They do contain, this is the phosphate, and this is, so there is the hydrophilic part, and then this is the hydrophobic part. They contain hydrophobic, no, phosphate is hydrophilic. That is, so how can a hydrophobic be soluble in water? So this is wrong. Then we come on to question number 12. Then question number 12, hydrogen bonding explains many of the properties of water, including the high latent heat of vaporization and high specific heat. So it says hydrogen bonding explains these properties. Now it says for which processes in plants is hydrogen bonding in water important on hot sunny days? Now, why is the answer B? 1, 2 and 4 only. Preventing denaturation of enzymes in the leaves. Hot sunny day, they'll keep it cool, so the enzymes will not be denatured. 2, reducing water loss by evaporation. It takes a lot of heat for it to evaporate. So less evaporation means less diffusion of water vapors. And holding the column of water in the xylem vessels together on a hot sunny day. This one is wrong because allowing leaves to cool down quickly at night. We are talking of a hot sunny day. We're not talking of the night. That was a good catch one. Question number 13. Which features of cellulose molecules contribute to the function of plant cell walls? Now, as I told you before, beta cellulose is made up of beta glucose, BC. So this is wrong. Alpha glucose. First, be very cognizant of this, that that's the ones which are wrong have to be removed immediately. Now you're left down with the choices of A, B, and D. So adjacent cellulose molecules are linked by glycosidic bonds. No, it says to the function of the plant cell wall. Plant cell wall is very strong. It doesn't burst. B, branch chains of beta are linked by hydrogen bonds. Unbranched chains of beta glucose are linked by hydrogen bonds. Now you must remember which is branched and which is unbranched. Cellulose is not branched. So that is why the answer is D, which is the unbranched chains are linked together. So there's one chain, there's another chain, and then it is attached by hydrogen bonds. That gives cellulose the strength. Read this up if you're not clear about this. Question number 14. Which statement describes some enzyme actions? Enzyme hold reacting molecules so that their reactive groups are close together. In an enzyme catalyzed reaction, more molecules have sufficient energy to react than without the enzyme. Reactions catalyzed by enzymes take place at a lower temperature than they would without the enzyme. You see, enzymes lower the activation energy. So that is why the answer is A. 1, 2, and 3 all are correct. Then coming to question number 15. Influenza virus has an enzyme called neuraminidase, which breaks down glycoproteins in the surface membrane of the cell that the virus will infect. The glycoprotein binds to the active site of neuraminidase by induced fit. Which statement about the induced fit hypothesis of enzyme action are correct? The active site must have the same shape as the substrate for them to bind together. The enzyme is less likely to be affected by non-competitive inhibitors than an enzyme working by the lock and key mechanism. The substrate is converted to product by specific R groups in the active site, just like the lock and key mechanism. Now, why is this wrong? The active site must have the same shape as the substrate. No. If this is the substrate is complementary to the active site. This is the substrate and this is the enzyme. So it is not the same shape. It's complementary. So this is wrong. This enzyme is less likely to be affected by non-competitive inhibitors than an enzyme working by the... That doesn't make any sense. So that is why the answer is D. Only this was correct. Now, question number 16, the graph compares the effect of temperature on the activity of the protease enzyme papain. So this is the free papain and this is the immobilized papain. So you can see the immobilized papain, what is happening to it. It is at this one, of course, is starting to decrease here at this temperature, but this one continues working. It has continued to work and is here. So it is going through a higher temperature. You can see the temperature here. So what is it doing? It increases the stability of the papain at higher temperatures. So it alters the shape of the papain's active site at higher temperature. No, doesn't seem so. It decreases the activity of papain at high temperatures. No, that's wrong as well. It reduces the number of collisions. No, we can't see that anywhere. Question number 17. The diagram shows how adrenaline activates processes inside a cell. You can see adrenaline, there's a beta adrenergic receptor, then there is cell surface membrane, then the G protein activates processes inside the cell. Now, it says which row is correct. The type of receptor molecule is a protein. Receptor changes shape. So, very easy. Was B. Question number 18. The statements are comparison of endo and exocytosis. Endo means taking in, exo means exit, taking out. Both mechanisms involve vesicles or vacuoles in the transport of materials across the cell membrane, correct? Both mechanisms occur to allow bulk transport, correct? Endocytosis involves taking materials into the cell. Exo means out, release of materials from the cell, correct? Some of the cell surface membrane is lost. Yes, because when there is going to be, this is like this. So some of the cell membrane is lost when endocytosis occurs, and there is an increase in the cell membrane when exocytosis. Exocytosis means, that this is the cell membrane and something comes here and comes attached here, so that will result in this becoming part of it then. So D, 4, all of them are correct. How many statements are correct? 1, 2, 3, 4, all of them are correct. Question number 19. A student was asked to calculate the surface area to volume ratio of an agar cube with a side length of 5.5 millimeter. Which row is correct? Of course, the answer is D, 1.1 ratio 1. I'll just show you how this was done. This area is length into breadth, so 5.5 into 5 point, and there's 6. It's a cube. And the volume is length into breadth into width, so it's 5.5. So this is 181 ratio 166. This is 1.1 ratio 1. Which statement about the behavior of chromosomes during telophase is correct? Question number 20. Now, the telophase, they become two. You see, one cell in telophase form two nuclei. Chromosomes attached to the spindle fiber at the equator. No, that's wrong. Chromosomes start to uncoil. Yeah, that's correct. Why C wrong? Chromosomes move to the opposite poles. No, in telophase, two nucleuses have been formed. Then chromosomes condense into compact structure. That's in prophase. Then coming to question number 21, the transmission electron micrograph shows a cell in a stage of the mitotic cell cycle. Which statement explains why it is difficult to identify the stage of the mitotic cell cycle? We don't know whether this is telophase or it is the end of anaphase. Why is it difficult? So chromosomes may have supercoiled and are visible, but centrioles are not visible. Anaphase may be continuing or telophase may be starting. Anaphase may be continuing or telophase may be starting. Yeah, you can see here, these are starting to condense here. So is it anaphase or is it telophase? Yeah, the answer is B. Why is C wrong? It is unclear whether the electron micrograph showed two cells in metaphase. No, metaphase, they have to align in the center. Some people may consider interphase to have started. Interphase, you can't see anything. Interphase, they are thin thread-like structures, the chromosomes. You can't sense, they haven't condensed. Question number 22. It says during lagging strand replication, this is the lagging strand which I've drawn in yellow. Short fragments of DNA are produced. The fragments are joined together by DNA ligase. Which row correctly describes the structure of these short fragments of DNA, which are called Okazaki fragments? They have base sequence that are complementary to the newly synthesized leading DNA strand. They have base sequences that are complementary to the newly synthesized leading DNA strand. Consists of one polynucleotide strand. Contains only carbon, hydrogen, oxygen, and nitrogen. Why? What about the phosphate? There's nucleotides, deoxyribose, an organic nitrogenous base, and a phosphate, PO4. So the answer is D. Question 23. The genome of the bacterium E. coli has been altered to enable it to code for an amino acid that is not found in nature. All the ATC DNA stop triplets on the strand of DNA that is transcribed have been substituted to ATT. The ATC triplet can then be inserted to code for the new amino acid. A new tRNA can then be constructed to carry the new amino acid. What is the anticodon of this new tRNA? So DNA is ATC, mRNA is UAG, tRNA is AUC. So the answer is B. 40% of the bases in a section of non-transcribed strand of DNA are purine molecules. What will be the total percentage of cytosine and uracil bases in the primary transcript that is transcribed from the other strand of the DNA? So we have 40% of bases in the non-transcribed strand are purine. Question number 24. 40% of the bases in a section of non-transcribed strand of DNA are purine molecules. I've just shown this to you. 40% non-transcribed strand. So the transcribed strand will have 40%. This will be the transcribed strand. That will be 40% T and C. And 60% will be A and G. Now it says what will be the total percent of cytosine and uracil in the primary transcript. So cytosine and uracil are only going to come here. Cytosine and uracil. So there were 60% A and G. So primary transcript means a pre-mRNA. The pre-mRNA is made from the main strand, the transcribed strand. They started with the non-transcribed strand, then you have to go to the transcribed strands, and then you have to figure it out. So 60% were A and G. So 60% will be U and C in the RNA. Because for every A, there's no T in the mRNA. This is the RNA. So primary transcript is the pre-mRNA. Primary transcript is the pre-mRNA. So now the number of 60 A and G would mean 60% U and C. Question number 25. Which statement about xylem vessel elements is correct? The answer is B. Pits enable the movement of water into adjacent xylem vessels. But why the other wrongs? Hollow vessels enable the constant movement of water up and down. That's wrong. In xylem, it is not up and down. In phloem, in some of the tubes, it's up and some down. It's bidirectional. Vessels contain numerous mitochondria. No, the vessels don't contain any, they're hollow. Cell contents die. Vessels contain C plates. No, that's in the phloem, it's not in the xylem. Question 1 to question 25 completed.